did like a bunch of labs in New Hampshire, and we were asked to present on one of them. The one that we got was gene transfer into plant cells using the gene gun. I'm Monique Honeyghan, and she's Delfija Pantovic. Yeah.
Sorry we don't have a PowerPoint presentation. We just, have some pictures and we're going to explain what we did inside New Hampshire.
Okay. The objective of the lab was to transfer the genes with blue traits into the cells of a popular plant. The blue gene was used so that the results of the gene transfer would be noticeable when the cells grew. We could have used the green fluorescent protein instead of the blue gene to make the plants fluoresce instead. The popular plant was used because it is a fast grower. She's going to explain the procedure.
The first thing we had to do was clean the DNA, in other words the blue gene, and coat it with gold particles. We used the gold because it was heavier and what's called more pure and a natural substance.
It was non-toxic.
It was not toxic to the plant also, so therefore it wouldn't die during experiments. This was already done for us, so we put it on a plastic rupture disc, and this disc, when we put inside the gene gun, would eventually explode, therefore transferring the DNA onto a petri dish.
Hold on a second. You remember that presentation with the melanoma, and they showed us the gene gun? Well, the one that we use, it doesn't actually look like a gun. It's like a chamber. So.
Yeah, because they use that one for mammals while we
You can see the picture.
Yeah, it's at this corner. Isn't that?
It's right there. We're going to show you more pictures.
We fastened the corresponding metal cover onto the roof of the gene gun and put the tile onto 2 slides lower under the metal cover where the 2 metal pieces, where they're almost touching. When everything was set up, the door of the gene gun is closed and vacuum sealed. So I was talking about when the rupture disc explodes, when it explodes, it goes through the stopping screen, which is basically so that the plate that we use doesn't go onto the petri dish, just the genes are transferred onto it.
Okay, did everybody understand that? Any questions?
What was the name of your experiment?
Gene transfer into plant cells using the gene gun. I'm sorry we don't have a PowerPoint, so if you want me to repeat anything, I can.
You said something about popular plant. Can you repeat that?
Well, we're trying to get genes into the popular plants because we're doing genetic engineering. So we use the cells of a popular plant because it will be easy to grow. Back to why we did all this. If the popular cells were allowed to grow, we didn't have enough time to watch them grow or something. So if they're allowed to grow, they will grow into popular plants. And the plants would have the new gene that we put in it.
The reason why we did this is because practically all the plants you get at a supermarket, such as tomatoes, are genetically modified. Because before, back in ancient times, tomatoes, they were poisonous to humans. So we had to genetically modify them so that because we knew that they would be tasty because we saw other animals eating them. So what we did was that we modified them so that we could one day eat them, and that's what we're doing right here.
Except we're not using tomatoes.
Yeah.
So the pictures.
So if the cells are allowed to grow into plants, will the plants be blue or should I use white?
I don't really know if they would be totally blue, but they'd have some blue.
Yeah, we have to first watch them grow, which might take a couple of weeks.
Okay. Pictures. That's Delfija doing the, she has the,
Putting the gold.
she has the disc that she's going to put in the holder. The holder's over here. And the other part of it is what we put in the top of the gene gun. Those are the discs again. Right there. Yeah, they have on each disc. You can see it. Well, you can't see it, but we could see the gold dust on it, and on each like gold particle there's DNA on it.
Yeah, the objective was to make sure that DNA would be completely wiped off the disc and onto the Petri disc.
That's what the gene gun looks like. Yeah, we were not excited to know that it was not an actual gun. So we put like the other like what do you call it? The holder. It has like another part to it, and we put it up here.
You can see like little slots on the side.
Yeah, we had to like screw it in, and then on a tile we have the holder, and we put it in there. And inside the holder is the rupture disc. So we put the tile right here. So then it would be like almost touching right here.
And then when the rupture disc exploded, the DNA would just go straight down and hit the Petri disc.
That's the Petri dish. This is after we did the experiment.
So you can kind of see a little bit of the gold on there.
If you can see blue, you're really, really good. But we can see blue. After we did the experiment, we had to like observe it. Like after like a couple days, we had to observe it to see the blue particles. You can't see them, but more pictures. That's me. Like we explained before, we put one tile here and the other one here. On this one is the Petri dish with the cells. The helium pressure is gonna come from here and come right down and like rupture the disc so that the gold particles get into the plant cells. That's it. Any more questions? Okay, Brian.
How does the gene gun work?
How does
You just close the door and it just does it by itself?
It has pressure control, so after we vacuum
You
seal
kind
the
of
door.
see them go to red at the top. So
Yeah.
we would close the door and then we press a button and it would vacuum out all the air. And then we press another button when it hit like 20 psi. Pressure, and then that would make the rupture disc explode and put the genes on there, and then we'd have to shut it off, vacuum it out again, and then take it out.
Any more questions?
No, I don't.
Questions?
How do you say the gene gun and the Petri dish because of the material they use?
The Petri dish, the gene gun, the microscope, what else? The rupture disc with the gold particles.
The stopping screen so that the gold plate when it go down to the particles.
Stopping screen.
Not gonna stop.
Stopping.
Okay, any more questions?